Good morning. In my previous class, we have started discussing biomolecules. As I have told you, there are four components of it. One is carbohydrates, second is proteins, third is your vitamins, fourth is nucleic acids. In my previous class, we have discussed regarding carbohydrates. Today we will be going to discuss the second one, that is protein. Protein forms the major proteins. It forms the major chemical components of cell. Next to water. The main constituent of cell is your protein. Now structurally, protein is a high molecular weight compound, consists of a large number of different amino acids. Nearly 20 different amino acids are required for the synthesis of protein. Now, the amino acids which body cannot prepare, it is supplied externally, are called essential amino acids. Now, so far the name of amino acid is concerned, it consists of two functional groups. One is amino, that is NH2 group. Another is acid group, that is carboxylic acid, that is COOH. Few examples I am giving under this head. You can see this compound contain both amino group as well as carboxyl group. Its name is glycine, GLYCENE. Another example I am giving you, CH3, NH2, COOH. Its name is alpha alanine. One more example, you can take CH2, COOH. This is your beta alanine. So you can find amino acids contain two functional group. One is amino group and another is carboxyl group. I have given you three examples: glycine, alpha alanine, beta alanine. Now what happens? Now during protein synthesis, amino acids combines with each other. Say, I am taking one of the amino acids, NH2, CH. Say here is a substituent or maybe hydrogen. And another amino acid, say CH, R2, COOH, where R1 and R2 may be hydrogen or some substituent or maybe some substituents. Now what happens when one of the amino acids having carboxyl group reacts with the amino group of another amino acids? There is elimination of one molecule of water, forming a linkage called CO and H linkage, where you can, or this is called peptide linkage. So first it forms peptides. Now you can notice this peptide compound contains also free amino group and free carboxyl group. Therefore, it again react with amino group of this part, can react with the carboxyl group of another amino acids, and the carboxyl group of this part also react with amino group of another amino acids, forms a long chain called polypeptide. This is called peptide. Now it forms polypeptide. So a long polypeptide chain thus formed is a protein molecule. So two amino acids combine to form a peptide where there is a CO and H linkage after elimination of one molecule of water. That peptide compound contains free amino group as well as free carboxyl group, which again react with other amino acids, forms polypeptide. Polypeptides combine, or polypeptide chains formed now constitute what is called protein molecules. Proteins are of two types. Number one, called fibrous protein. Fibrous protein. This type of protein consists of linear thread-like molecules which remain side by side to form a fibrous protein. The molecules are held by what is called hydrogen bond. Hydrogen bond. They are usually insoluble in water. Some common examples of fibrous protein are your bit1, keratin, this is present in skin, hair, nail and wool. Keratin. The second types of fibrous protein is your collagen, collagen. This is present in tendons. Third example is your fibrin. It is present in silk. Another is myosin. It is present in muscle. So fibrous protein, as I have told you, consists of linear thread-like molecules which remain side by side, forms fibers. Fibrous protein, the molecules are held by hydrogen bond, and these are usually insoluble in water. Now this type of protein serves as the main structural material of animal tissues. The second category of protein is your globular protein. Globular second category. Now, in this type of protein, the molecules are folded together into a compact unit having spherical in shape. Here also the peptide chains are held together by same hydrogen bond. Remember, in both the cases, fibrous protein as well as in globular protein, the peptide chains are held together by hydrogen bonds. Now, these are soluble in water, and in aqueous solution they are soluble. These are soluble in water. The fibrous one is insoluble, also in aqueous solution of acid, bases and salts. These are soluble. Common examples are enzymes and some hormones like insulin. So, enzymes, which are also called biocatalyst because they act as a catalyst in different biological activities. Enzymes, otherwise called biocatalyst. And some hormones like insulin belongs to this globular protein. Next we shall discuss what is called denatured protein. Denaturation is a process by which we get denatured protein. The process that changes the physical and biological properties of protein without affecting its chemical composition. It only changes the physical properties and biological properties. Without changing its chemical composition, it's called denaturation. That process is called denaturation. It is usually affected by change in pH. If we change the change in pH, or by applying pressure or applying temperature change, and in presence of some salts and certain chemical reagent also. Chemical reagents are responsible for the denaturation of protein. We can change the physical property as well as the biological property without changing its chemical composition, either by changing pH or by changing temperature in presence of some salts or some chemical reagents. That process is called denaturation. Next we shall proceed how to know the presence of protein, that is test for protein. Now, on adding a dilute copper sulfate solution to an alkaline solution of protein, add copper sulfate solution to an alkaline solution of protein, you will get a violet color, colored compound due to formation of biuret. So therefore this test is called biuret test. biuret test. Next we shall proceed to the digestion of protein. So to know whether protein is present or not, add copper sulfate solution to an alkaline solution of protein, when you will get a violet color, that is due to formation of biuret. Therefore this test is called biuret test, which indicate the presence of protein. Now during digestion process, digestion of protein, we take protein. Now this is first reacted by your gastric juice, that is in the stomach, gastric juice, when it breaks to polypeptides. Now these polypeptides reacted by pancreatic juice, when it breaks to peptides. Peptides react by a biocatalyst, that is peptidase, when it breaks to amino acids. Amino acids can be absorbed by intestine, small intestine. So we take protein, acted by gastric juice gives polypeptides, which reacted by pancreatic juice gives peptide. By the action of peptidase, it gives amino acids. So this proves that amino acids are ultimate constituents of proteins. So amino acids, mostly alpha amino acids, are responsible for protein synthesis. Next I am going to give the uses of protein. Uses. Now number one, so far the uses of protein is concerned. Protein constitute one of the important food material, one of the important food materials for animal life. Second, your wool and natural silk, which are used for fabrics, are protein in nature. Third, proteins like hemoglobin, and various peptide hormones are essential for different metabolic activities of the human system. Fourth, we can find various enzymes, as I have told you, enzymes essential. These enzymes are essential for different transformation, but different chemical reaction inside the body are also protein in nature. Many antivirus vaccines are also proteins in nature. That is in the form of, we use this in the form of denatured protein, in the denaturation form. Sixth, we find like glue or your casein or gelatin, which find industrial applications, are also protein in nature. Protein are also sources for different amino acids. Amino acids are required for different biological function of human life. So, it important uses, it constitutes one of the main constituent of food materials for animal life. Wool, natural silk are also protein in nature. Hemoglobin and some peptide hormones which are used or which are necessary for different biological function are also protein in nature. Enzymes which are act mostly as a biocatalyst are also protein in nature. Antivirus medicines are mostly denatured protein. Glue, casein, gelatin which are used industrially are also protein in nature. Amino acids are essential for different body functions are also protein, or we get amino acids from protein. So, this is all about protein, mostly asked as a short note. So, if it asks short note, you will have to write everything. And the main constituent of amino acids is your protein is alpha amino acids, and sometimes ask test for protein, sometimes ask short note denatured protein. Next I am going the third category of biomolecules, that is vitamins. Now these are the organic compounds which cannot be produced by body. It cannot be prepared by the body and must be supplied in small quantity in the form of diet, that is in the food, for the maintenance of normal body health. So I am repeating so far. If you can, what are vitamins? Now these are the organic compounds which cannot be produced by the body. It must be supplied in small quantity in the form of diet or in the form of food for the maintenance of normal body health. Their deficiency causes some diseases, particular diseases. Chemically, their composition is very complex, but in short, we abbreviated them as vitamins A, B, C, D, E and K. So these are the different types of vitamins which body cannot prepare. It is supplied in smaller quantity in the form of diet or medicine or food, whatever you can call it. Now these are two types. One is water soluble vitamins. Under this head comes vitamin B and C. And the second one is fat soluble, dissolved in fats. Fat soluble vitamins, examples are A, D, E and K. Now I will discuss different source for different vitamin, different sources, and the deficiency caused in its absence. Most of the time ask as a short note or fill up the gaps or multiple choice. So vitamin A, the sources are, or from where you can get it, sources, your butter, ghee, liver, carrots, spinach, kidney, yellow pumpkin, milk, fish, egg yolk, your cheese, papaya, mangoes, etc. These are the different sources from which you can get vitamin A. Disease, that is disease caused in the deficiency of vitamin A. Disease caused due to its deficiency is night blindness, and cirrhosis. So, short note sometimes ask 2 marks, what are its sources of vitamin A and what disease caused due to its deficiency. Next is vitamin B. It contains 4 parts: B1, B2, B4 and B12. Let us start vitamin B1. Sources is your milk, pulses, wheat bran, seafood, yeast, green vegetables, cereals, etc. So these are the different sources where you can get vitamin B1. The disease caused due to its efficiency is very. The next vitamin is vitamin B2. Vitamin B, I have told you, divided into 4 parts: vitamin B1, vitamin B2, vitamin B4 and B12. Vitamin B2, the sources are your meat, pulses, milk, yeast, liver, peas, egg and green vegetables. The disease caused due to its efficiency is cracking of skin, fatigue, cracking of skin, lips and corner of mouth, and photophobia. Photophobia means roughness of eyelids. Roughness of eyelids. So the sources are meat, pulses, milk, yeast, liver, peas, egg, green vegetables. The disease caused due to its deficiency, cracking of skin, lips, corner of mouth, and photophobia, that roughness of the eyelids. Next is vitamin B4. Vitamin B4, source from which you can get vitamin is milk, egg, meat, fish, green leafy vegetables. These These are the different sources where you can get vitamin B4. The deficiency of vitamin B4 causes the disease called pellagra. Pellagra. So different sources where you can get vitamin B4: milk, egg, meat, fish, green leafy vegetables, and the disease caused due to its deficiency is pellagra. Next is vitamin B12. Vitamin B12. These sources are you can get vitamin B12 from milk, egg, fish, meat, fish, liver, cheese, cheese, etc. These are the different sources. You can get it from milk, egg, meat, fish, liver, and cheese, etc. And the different disease caused due to its absence is inflammation, inflammation of tongue and mouth. This is one disease. Second, you will get pernicious anemia. So due to vitamin B12 absence, there is inflammation of tongue and mouth and pernicious anemia. You can get vitamin B12 from milk, egg, meat, fish, liver, cheese, etc. Next is vitamin C. Vitamin C mostly you can get from amla, tomatoes, oranges, that is citrus family fruits, lemons, peach, pineapple, cabbage, apples, green chili, lonka, guava, etc. These are the main source from which you can get vitamin C. That is amla, tomatoes, oranges, lemons, peach, pineapple, cabbage, apples, green chilies, and guava, etc. The disease caused due to its absence is scurvy. Next is vitamin D. Vitamin D. The different sources where you can find vitamin D or which provide vitamin D to our body is cod liver oil, butter, milk, egg yolk, fish, ghee, cheese, etc. And another important thing also, exposure to sunlight, morning sunlight mostly, exposure to sunlight also produces vitamin D in our body. So source is you can get from cod liver oil, butter, milk, egg yolk, fish, ghee, cheese, etc. And morning walker, that is when they exposed to sunlight, it also helps in the preparation of vitamin D in our body. Deficiency of vitamin D causes rickets. Next is vitamin E. Vitamin E. The different source from which you can get vitamin E is your vegetable oil, milk, tomatoes, green leafy vegetables, egg, kidney, cereals, nuts, etc. These are the different sources which provides vitamin E. The disease caused due to its efficiency is loss of reproductive capacity. So different source which provides vitamin E is vegetable oil, milk, tomatoes, green leafy vegetables, egg, kidney, cereals, nuts, etc. Due to its efficiency, there will be loss of reproductive capacity. The last one is vitamin K. Vitamin K. Now, different source from which you can get vitamin K is same green leafy vegetables, cabbages, soybean, and vegetable spinach and tomatoes, etc. Deficiency of vitamin K causes lengthening of blood clotting time, lengthening of blood clotting time. It also causes hemorrhage. So this is all about vitamins, where I have told you what are the different sources of getting vitamin A. B, there are 4 spots: vitamin B1, B2, B4, B12, vitamin C, vitamin D, vitamin E, and vitamin K, and different disease caused due to its efficiency. Next we shall proceed to under this biomolecules is your nucleic acids. Nucleic acids, D part. Now, as you know, proteins and enzymes have their importance in carrying out different or various cellular functions. The different function carried by cells is done by proteins and enzymes. Enzyme is also a protein in nature. Now, the role of nucleic acids, that is functions of nucleic acid, is how these molecules originate in the cell, originate in the cell. And second is, how do these cells preserves the structural informations, structural informations. So, you know, in the cell, the main constituent of cell is your protein. They have got cells, protein and enzymes has got different functions carried by the cell. Now, nucleic acid provides how these molecules originate in the cell and how does the cell preserve the structural information. So, for the definition of nucleic acid is concerned, nucleic acid is defined as nucleic acid is defined as the long chain polymer. It is not a simple molecule, it is a polymer present in high concentration, in the nucleus, plural is in the nuclei of the cell. Hence the name is nucleic acid. Cells in which the monomer, since it is a polymer, so monomer units are nucleotides. So once again I am telling you, nucleic acids are defined as the long chain polymer. It is a polymer of the monomer nucleotide. Polymer present in high concentration in the nuclei of the cell, in which the monomer unit is on nucleotides. Now, different components or different compounds present in nucleic acid, that is, we can write as the constituents of nucleic acid. What are its constituents? Constituents of nucleic acids. Number one, it contains phosphate unit, a phosphate unit, or that is a phosphoric acid moiety, moiety. The second, it contains a sugar. These are again two types. One is ribose sugar, another is deoxyribose. Third unit, it contains nitrogenous base, mostly heterocyclic bases, These are again two types. One is called purine bases. Under that head, it is adenine, another is guanine. And second type of base, that is heterocyclic compound where nitrogen is in the ring, is pyrimidine bases. Pyrimidine bases. It can be cytosine or it can be thiamine or it can be uracil. So the main constituent of nucleic acids are three types. One is phosphate unit, that is a phosphoric acid moiety. Sugar, that can be again two types, it can be ribose sugar or deoxyribose. Third one is a nitrogen bases, that is heterocyclic bases. These are again of two types. One is purine bases, it can be adenine or guanine, or pyrimidine bases, not or, it contain also pyrimidine bases, it can be cytosine, thiamine and uracil. Now, as I have told you, nucleic acids is a polymer of nucleotides. So first we shall see now what is nucleotide, structure of nucleotides, from which or nucleotides polymerizes to form nucleic acid. Now structure of nucleotides is like this. Structure of nucleotides. In nucleotide, the sugar ring is attached to sugar, attached to the nitrogenous bases through its carbon atom 1. This this bond is called glycosidic bond. So sugar combines with the nitrogenous base through its first carbon atom, C1 carbon atom, and this linkage को कहते हैं bond known as glycosidic bond. Now the phosphate group is linked to the hydroxyl group of the sugar. Now we can see the sugar unit, sugar combines with a base, again it combines with the phosphate of another nucleotide. Again it combines with the sugar unit, sugar combines with a base, again a phosphate, again sugar, where sugar combined with a base. So thus nucleic acid backbone consists of alternate sugar and phosphate residue, and one of the four bases, I have written 5 bases, 2 of them are purine bases and 3 of them are pyrimidine bases. So only 4 of them are present in your nucleic acids, which I am now giving you types of nucleic acids. There you can see only out of 5 bases we get, or there is only use of 4 bases. They are types of her types is concerned, that is one is called DNA bit 1 types. DNA, that is deoxyribonucleic acid. And now this DNA have large molecular mass, that is starting from 6 to 16 million, and are found primarily in the nucleus of the cell. Nucleus of the cell. And it is responsible for the hereditary information from parents to the offspring. The second category is your RNA, that is ribonucleic acid. Now, these are of less molecular mass, from 20,000 to 40,000. 20,000 to 40,000, and are found mostly outside the nucleus, that is inside the nucleus, is outside the nucleus, that is in the cytoplasm. Cytoplasm. The RNAs are of 3 types again. Sir, RNA of 3 types. One is called messenger RNA. So in short we write mRNA. These are they transmit genetic information from DNA molecule to cytoplasm. The second one is your transfer RNA. Or in short written tRNA, and it acts as an adapter which carries the amino acid to the growth of the polypeptide chains. The third one is ribosomal RNA, or called RNA. It forms an integral part of the ribosomal structure. Next I shall discuss. So there are 3 types. One is messenger RNA, transfer RNA, and ribosomal RNA. Distinction between or difference between DNA and RNA. Number 1, in DNA the sugar unit is 2-deoxyribose sugar, and in RNA the sugar unit is ribose. The second, now in DNA the base thiamine is present, but not uracil. In RNA, base uracil is present, but not thiamine. Third, DNA is a double helix structure, double helix strand. Here it is a single strand. Fourth, DNA present in the nucleus. Fourth point, DNA present inside the nucleus. Here it is present in the cytoplasm, that is outside the nucleus. Fifth one, it is responsible for hereditary information from parents to the offspring, from parents to the offspring, hereditary informations from parents to offspring, and RNA is responsible for protein synthesis. So, as I have told you, nucleic acid is a polymer of nucleotide. There is another word called nucleosides. Nucleosides is obtained when nucleotides are divided of phosphate unit. So, nucleic acids are two types, DNA and RNA. So, DNA, the sugar present is 2-deoxyribose, here is ribose. Second, in DNA the base thiamine is present, not uracil, but RNA, uracil is present, not thiamine. DNA is a double helix structure, it is a single strand. DNA present in the nucleus, it is present in cytoplasm. DNA carry the hereditary informations from parents to offspring, but it is mostly responsible for protein synthesis. So, this is all about the nucleic acids. So, we have completed the discussion of biomolecules like carbohydrates, proteins, vitamins and nucleic acid. Next class, I shall have to discuss a quite large number of things, that is polymers and chemistry in the service of mankind. Thank you.